Hello everyone. So now we are going to discuss amongst the next in the series of the water soluble vitamin, biotin. It's also goes by the name B7, and it was earlier also called as vitamin H. That is from a German word called Haar and Hart, which in German means hair and skin. This vitamin was discovered by an American biochemist, Dean Burke. Let's see the structure of this particular vitamin, biotin. So you could see that this particular group, this NH, CO and NH, that's called urido, and which is a tetrahydroimidazolone group, and linked to a tetrahydrothiophene part, and which is again linked to an aliphatic carboxylic acid chain, that's valeric acid. And the active form of biotin is actually the biocytin, which is attached to eta amino biotinyl lysine. Like this lysine group is actually linked to a protein molecule, and the protein which is having this lysine is protruded out, that is linked to the carboxylic acid chain of this valeric acid. So on the proteolysis, or when the proteins are degraded, this biotin is actually released and utilized by the system. Now let's talk about the biological importance of the biotin, or you can say what kind of reactions actually this biotin catalyzes, and that's very long list. So first of all, let me just write it down, that list. Biochemical importance of biotin. Biotin that is in the D form is used for as a coenzyme for carboxylase enzymes, that is for the carbon dioxide transfer reactions. So it is utilized in the synthesis of fatty acids and the catabolism of the amino acids, like branched amino acid like isoleucine and valine, and it's also involved in gluconeogenesis. And some of the carboxylase enzymes that utilize the biotin as a coenzyme are, let me just write it down. Biotin is a coenzyme for the carboxylase enzymes, which I told you earlier that these are needed for the carbon dioxide transfers. And some of the important carboxylase enzymes are the alpha acetyl CoA carboxylase and beta acetyl CoA carboxylase, and another one methyl crotonyl CoA carboxylase and pyruvate carboxylase and propionyl coenzyme carboxylase. So these enzymes are actually responsible for synthesis of the fatty acids and the catabolism of isoleucine and valine, and they are also involved in the important process of synthesis of glucose, that is gluconeogenesis. Let's move on to the next page and talk about the deficiency symptoms associated with biotin. Biotin is one of the enzyme which deficiency is rare because it's available in wide-ranging food, but the deficiency could arise as a result of the loss in the bioavailability. Bioavailability of biotin is low, especially like 20 to 40% in the grains or cereals, and the alcoholic have the low biotin circulating level in the system. The deficiency of biotin can lead to the brittle and thin fingernails, as the name earlier we talked about. It's also called as vitamin H, that is for in German Haar and hot, which means hair and skin. And the another symptom is hair loss or conjunctivitis, dermatitis. It can lead to the neurological problems also, like the depression, lethargy, and hallucinations. And another very interesting symptom is numbness and tingling in the extremities, that is in the hands or the feet. Rich source of vitamin B7 or biotin are the leafy green vegetables, peanuts, cheese, raw egg yolk, and liver. A very important thing that is actually observed in the pregnant women, if there is deficiency during the pregnancy days, it can result in the congenital deformation like the cleft palate in the newborn babies. The other important usage of vitamin B7 or biotin are this particular vitamin biotin have a very strong binding in the biological word known to till date, that is with a protein present in the egg white that is called avidin. Let me just write down a little bit for you. Biotin is actually known to form very strong non-covalent binding with a protein present in the egg white, that is avidin, and its dissociation constant, that is 10 raised to the power minus 15 mole per liter, and another tetrameric protein, streptavidin, present in a microorganism or isolated from this microorganism called Streptomyces avidini, which a comparative dissociation constant of 10 raised to the power minus 14 mole per liter. So it is one of the strongest known covalent interaction between a protein and a small molecule, that is biotin. So it results in the egg white injury also. That is, if suppose somebody for very long time is relying on the egg whites for a source of nutrition, that can actually result in the deficiency of biotin. So these two very strong interactions are actually be utilized in biotechnology for the purification. So this particular interaction of biotin with these proteins, avidin and streptavidin, is actually utilized for the purification of the proteins. Like in this particular process, the biotinylated proteins are actually incubated with the avidin or streptavidin. So as a result of this very strong interactions, the biotin and avidin, those proteins are actually fixed to the surface of the matrix on which this avidin is coated, and thereon later on these proteins or those proteins can be purified in the pure form. And the another interesting use of biotin is in the ELISA, that is enzyme linked immunosorbent assay, where the secondary antibodies linked with the biotin are utilized for capturing the primary antibodies, and which can later on be utilized for identifying by using streptavidin conjugated with a reporter molecule like horseradish peroxidase or alkaline phosphatase.